Welcome to this explainer. Today, we're going to embark on an absolutely incredible journey using recently declassified expedition logs from the interstellar research vessel UNS-7. We're going to completely unravel the discovery of an alien ecosystem, one that operates on a totally different set of chemical rules than anything we've ever seen before. It's a story of survival, of chemistry pushed to its absolute limits, and honestly, of a shadow biosphere that completely redefines what it means to be alive. Let's get right into it. Okay, let's dive into this by asking a pretty fundamental question, one that really challenges our terracentric dogma. Could a thriving, complex ecosystem exist entirely without phosphorus? Now, for decades, astrobiologists believed the answer was just a definitive no. On Earth, as you know, phosphorus is the undisputed cornerstone of life. It's the structural backbone holding our DNA together, and it's the vital energy currency pumping through our cells. Taking phosphorus out of the biological equation, well, it's kind of like trying to build a skyscraper without steel. It's the ultimate biological rule. But we thought we had tested this rule before. Back in the early 21st century, scientists found a bacterium named GFAJ1 out in California's Mono Lake. For a brief, electrifying moment, the whole world thought we'd found an organism that actually swapped out phosphorus for its chemical cousin, arsenic. But that dream of arsenic-based life was totally crushed. Why? Because of water. Arsenic bonds, specifically arsenate esters, are catastrophically unstable in an aqueous environment. While Earth's standard phosphate bonds can chill in water for millions of years without enzymes, arsenate bonds violently hydrolyze. They literally break apart in just minutes. An arsenic-based organism in water would dissolve its own genetics faster than it could reproduce. So that firmly cemented the phosphorus rule in science. Arsenic is an imposter, water is life, end of story, right? Well, then the UNS-7 arrived at NYX-7. This is a tidally locked super-Earth orbiting a dim red dwarf star. But instead of our familiar oxygen-rich atmosphere, NYX-7 is absolutely choked with nitrogen, argon, and traces of highly toxic arsine gas. And instead of blue water oceans, it features deep, viscous, violet oceans of a chemical called Formamide. The shorelines, they aren't made of sand, they're these jagged formations of arsenic sulfides like orpiment and realgar. The temperature holds super steady at a chilly 12 degrees Celsius right at the terminator line. Make no mistake, to anything from Earth, this entire planet is basically just a giant vat of toxic waste. Which brings us to the central scientific mystery of the expedition, and it was perfectly captured by the mission's geochemist Dr. Julian Mercer. He pointed out that a formamide ocean is an absolute nightmare for cellular biology. The reason? Well, standard Earth cells are encased in a lipid bilayer. If you drop our cellular membranes into pure formamide, poof, they simply dissolve. The planet literally shouldn't be able to support life as we know it, and yet, the SAGN's long-range sensors were picking up massive, undeniable signs of biological activity. So the big question was, how on Earth, or rather, how on NYX-7, could life survive in these viscous violet seas? The answer lay in the Kylos ecosystem. It's this environment of terrifying, toxic beauty, strictly confined to the twilight zone of this tidally locked world. The SAGN team discovered these metallic, golden ferns that look like spun glass, actively absorbing the weak infrared light from the red dwarf and venting arsenic gas. Clinging to the arsenic rocks were these lichen-like crusts pulsing with a sluggish, bioluminescent green glow. And beneath the violet waves, glass crawlers. These benthic, trilobite-like creatures with totally transparent arsenic sulfide exoskeletons. You could actually see their dark purple, formamide-based blood pumping right through their visible veins. It was a complete, thriving biosphere. But what in the world was it made of? The Kepler descent team took a sample of that golden fern into their mobile lab, expecting to find, the standard building blocks of life. Instead, the mass spectrometer returned a truly shocking number: 0. Absolutely 0 phosphorus was detected, literally not a trace. Instead, the intracellular concentration of arsenic was a staggering 2 molar. To put that into perspective, a concentration that impossibly high should be instantly lethal, yet the impossible was staring them right in the face.
Now, what's really interesting here is seeing exactly how life on NYX-7 actually pulls off this impossible chemistry. On Earth, our DNA uses a phosphate backbone as a sturdy bridge between nucleobases. But NYX-7 life uses DANA, deoxyriboarsenate nucleic acid. It completely substitutes phosphate with arsenate. Now remember the ghost of Mono Lake? Arsenate bonds are supposed to shatter in water. But the brilliant twist of NYX-7 is, there is no water. The intracellular fluid is pure formamide. Formamide is a fantastic solvent, but it lacks the highly reactive hydroxyl group found in water that destroys those arsenate bonds. In an environment totally devoid of water, an arsenic backbone is perfectly stable. So on NYX-7, water is actually the lethal poison.
And this biological substitution dictates their entire way of life. Just look at their energy currency compared to ours. Earth life uses ATP, adenosine triphosphate, which stores massive amounts of energy in tight bonds for explosive metabolic power. NYX-7 biology uses ATSP, adenosine triarsenate. The bonds between arsenic and oxygen are naturally much weaker. They require significantly less thermodynamic effort to form, and they release less energy when broken. So the crucial point is, why does this matter? Because NYX-7 orbits a dim, red dwarf. The star just doesn't provide enough high-energy photons to drive Earth-like photosynthesis. This entire biosphere relies on thermodynamic frugality. It's a low-energy ecosystem perfectly adapted to a low-energy star. This creates a perfectly closed shadow biosphere loop. Let's walk through it. It starts with the golden ferns using infrared light to oxidize arsenite into arsenate. The ferns then use that low-energy reaction to build ASTP, fixing carbon from the formaldehyde and venting arsine gas. Next up, the glass crawlers eat the ferns, using that arsenate to breathe instead of oxygen and finally reducing it back down to arsenite to complete the planetary cycle. It is a brilliant, delicate symphony of formaldehyde, arsenic, and infrared light. But understanding this beautiful system almost cost the crew everything, and it started with a terrifying lab accident they dubbed the 10 microliter catastrophe. Dr. Mercer was running a totally routine test on a fast-growing Nixine yeast. Out of decades of earthbound habit, he adjusted the pH using 10 microscopic microliters of a standard sodium phosphate buffer. The result was instantaneous. The thriving purple cells underwent catastrophic necrosis, basically massive and instant cellular death. Their internal transport vesicles froze up, and within 30 seconds, every metabolic process hit a literal brick wall. The entire colony was dead. To understand why a tiny drop of our life-giving phosphorus was a weapon of mass destruction to them, we need to look at the concept of a competitive inhibitor. It's basically a substance that wedges into an enzyme's active site and gets permanently stuck. To Earth life, arsenic is toxic because it mimics phosphorus, sneaks into our enzymes, and then breaks apart way too easily. But on NYX-7, the exact opposite is true. Phosphorus mimics arsenic, gets pulled into their low-energy enzymes, but the phosphorus-oxygen bond is just incredibly strong. Their biology literally doesn't have the thermodynamic power to cleave it. So the phosphorus goes in, gets permanently wedged, and acts as a massive metabolic wrench in the gears. Dr. Alara Vance, the chief exobiologist on the mission, summarized the sheer poetry of this discovery with this thought: "We are perfect chemical opposites, two mirrors facing each other across the periodic table." It's a profound realization, right? Our foundation is their poison, and their foundation is our poison. And while that's scientifically fascinating, this mirror image chemistry suddenly set up some incredibly deadly life-or-death stakes for the Kepler descent crew. Let's move to and see how this builds. Just 3 days before departure, this mirror image chemistry turned into an escalating disaster. A severe storm triggered an airlock breach. A 200-kilometer-per-hour wind caused a microfracture, violently blowing formamide and arsenic dust into the chamber, with Dr. Vance trapped inside. The ship's AI detected foreign biology and did exactly what it was programmed to do. It initiated a standard purge. It flooded the chamber with boiling water and hydrogen peroxide. But remember, water is lethal to DNA. The steam violently attacked the alien cells, causing their arsenate backbones to instantly hydrolyze and detonate. This runaway reaction caused a massive oxidation event, producing an explosive spike of lethal arsine gas right inside the airlock. The sensors spiked to a terrifying 4,000 parts per million of arsine gas. To put that into perspective for you, arsine gas binds to hemoglobin and instantly destroys human red blood cells. A concentration of just 250 parts per million is lethal within minutes. Dr. Vance was trapped in a chamber with 4,000 ppm, and this highly acidic gas was rapidly eating through the rubber seals of the inner door, threatening to flood the entire ship. And worse, the storm had jammed the outer door shut. They were literally trapped by their own sterilizer. With literally seconds to spare, Dr. Vance realized the only way to stop a runaway thermodynamic reaction was to rob the room of all its energy. Bypassing all safety protocols, she plugged her suit's sample-preserving liquid nitrogen directly into the airlock's recycler. The room temperature plummeted to minus 80 degrees Celsius. The airborne formaldehyde droplets instantly turned to solid, inert ice. The deadly arson gas, starved of thermal energy, condensed into a toxic liquid and then froze solid onto the floor as a sheer white frost. Just like that, the chemical weapon was completely halted in its tracks. As the team safely returned to orbit, that near-fatal disaster left them with a sobering philosophical reflection on the uncompromising laws of physical chemistry. Dr. Vance noted in her final log, "Life is a thermodynamic process. It is a localized rebellion against entropy. You see, on Earth we fight that rebellion with fast, hot, high-energy phosphate bonds in an aggressive solvent called water. But on NYX-7, the rebellion is quiet, it's slow, and it's frugal, hiding in formaldehyde to protect fragile arsenic bonds. We can never walk on their shores without armor. Our biospheres are fundamentally chemically locked apart."
Please like and subscribe.